important to do, but it can be a little depressing, particularly if you're at this end of the age spectrum, which I am in. So, and on the other hand, as I was saying to Rita Brown earlier, this particular week.
No, sir. Sorry.
All right, well, it's all right. It's all right. We were waiting for it. It's actually, from my perspective, from the science side, far and away the most interesting. And it's just fascinating. And you'll see why I'm very excited about this one. There are probably at least 2 or 3 Nobel Prizes working in this material, future ones coming along. And you'll understand why and some of the implications as we move into this. Alzheimer's disease, we're talking about next week. And I added that one because I suspect that most people in our third of life, the last third, it's something we think about or we see in our friends or partners, some cognitive decline or behavioral changes, and it's scary, especially if we experience those things. So lurking underneath the surface for many is kind of concern about what seems to be an unstoppable disease, these neurodegenerative diseases, and there is some hope in that, and we're going to talk about that next week, but not yet funded by the Canadian government or the UK government as yet, and very expensive in the US. So that's what we'll talk about next week. The other thing, the Nobel series is coming up in early November, but this year I've decided to look at some other prizes as well. The Lasker prizes were announced this month. They're often a prelude to the Nobel prizes, with a gap of as little as 1 year, or sometimes 4 or 5 years. And so it's well worth it looking at other prizes in engineering, math, and that have matured. And yes, the Nobels have been around, and they set the standard, but in some years not so well. And so this is good competition, I think. So let's get into this stuff. I'll tell you, this sets the tone, I think. There's a wonderful book written by Philip Ball, How to Grow a Human, Adventures in How We Are Made and Who We Are. Absolutely. Not many books fit the title. This book fits the title. It's that exciting. And it covers a kind of field in science that's really been coming along quietly and a little bit more urgently in recent years. And we're going to talk about some of that. But in one sentence at the top, every part of our cells can potentially be turned into any other part of our cells, including a complete self. Wow. That means that every mature cell in our body could potentially be reverse engineered to become an embryonic stem cell, or the equivalent, and then redirected to become some other tissue. That's a huge change. It's already won 1 Nobel Prize, and more are certain to come. We tend to think of cells or bodies as this progression from birth, childhood, adulthood, early adulthood, and late. So beginnings to endings, so to speak. But biologically, it's not quite that way, as we're finding out. And therein lies considerable hope, I think, for the future. So let's just move along from, this is why I said here, there are Nobel Prizes and then there are game changers. And we're going to hear about potential game changers here. And 2 people who were game changers. And I couldn't resist this one. We've all been blessed by very good teachers, some average teachers, and some not so good teachers. And And the not so good ones can really hold back young kids. And so here's an example of that, about a fellow, John Gordon. His high school teacher said this: "I believe he has ideas about becoming a scientist. On his present showing, this is quite ridiculous. If he can't learn simple biological facts, he would have no chance of doing the work of a specialist, and it would be a sheer waste of time on his part and those who have to teach him." What a downer to get. Well, thereafter, Gordon was not allowed to take any more science courses and studied languages. Then what was unforeseen by the teacher happened. He got a Nobel Prize for exactly what we're talking about. And so what did he do? Well, this was the beginning of kind of the biological revolution of changing cell identity and making older cells new. And so he used ultraviolet light to destroy a frog egg nucleus, and then combined that with an adult, well-differentiated epithelial cell from a tadpole to kind of fill in the dead nucleus here. And lo and behold, guess what happened? That single cell developed into a tadpole, and followed enough into a frog. So the point is that every cell in our body, every epithelial cell in our gut, every heart cell, potentially almost every cell can be reverse engineered. I mean, remember that cells in the embryo, started out with a single cell, and then they multiplied, but they also began to differentiate from one another into becoming, brain cells, or into gut cells, or into heart cells, that kind of thing. So it followed that train that we're very familiar with. Well, we just reversed the train. The point is that mature, well-differentiated cells are quite capable of being re-engineered with the right transcription factors, proteins, that can turn them around or reverse them. And so there's the fellow who got that damning assessment from his teacher, and I would say he did rather well.
How did he do it?
Well, we can talk about that a little bit later, but well, here we are with Dolly. This is just another play on this. Transfer the nucleus from a mammary gland of an adult sheep. After taking out the nucleus from the sheep egg, so you're replacing the sheep egg nucleus with an adult nucleus from the mammary gland, from the breast, from which union a sheep was born. That was Dolly. Okay? So what did Dolly show? That mature somatic cells have all the genes needed to make a complex organism, and that they can become active again. Think about the potential of that. That's where the next 2 or 3 Nobel Prizes, or at least some of them, are going to come from. Playing on that very theme. That was an unimagined capability, certainly 30 years ago. And, but think about it. Egg cells have, well, here we are, but this is the question. But might a somatic cell's genes be reset without having to pull out its nucleus, stick in an empty egg, stick it in an empty egg, and grow it into an embryo? Well, that's a very cumbersome way of going about it. And this fellow, Shinya Yamanaka in Japan, he shared the Nobel Prize with John Gordon in 2012 for developing another way of going about this. And so what did Yamanaka do? he identified 24 transcription factors, read proteins, that were capable of reversing adult somatic cells into their much earlier forms that were capable of becoming any cell. And he piggybacked those transcription factors into adult cells by piggybacking them on the virus. So that's just using a virus as a vector to get those transcription factors into the cells. It worked. Adult somatic cells from an adult mouse fibroblast were induced to become these pluripotential, potent cells. And then he later showed he didn't need 24. Only 4 of the original 24 transcription factors were actually needed. And they christened these cells. In the literature, you'll see this all the time. iPSC cells, or induced pluripotent stem cells. So that's part of the language of this new field. And they reported their findings in 2006, and in 2012, he and John Gurdon shared the Nobel Prize in Medicine. And big understatement by the Nobel Committee here, paradigm shifted our understanding of cellular differentiation and plasticity. That is an understatement. It's much bigger than that. Well, and this is kind of what our Japanese friend did, just took a fiberglass from, say, the skin or any other source, but adult cells here, mixed them with the transcription factors, and eventually that transformed him into an adult mouse. Amazing, isn't it? It's actually incredible. And but also, it's interesting from a disease point of view, because you can also grow abnormal cells. So if somebody has a genetically transmitted disease or some other disease, then why not grow its cells, reverse engineer them, and see how that disease expresses itself in specific tissues? Follow what I'm saying now? Okay, it's important to follow the path here and see why I'm so excited about it. So you can actually redirect those iPS cells into nerve cells, heart cells, liver cells, any other tissue you want. So it's really an incredibly powerful way of getting at things and studying things. And that's only a decade ago. If you thought gene was a big thing, this is much bigger than gene editing. This is editing all the genes. Well, what about organoids? Can you make organs? And yes, you can. So these pluripotent stem cells can be coaxed into developing into the skin, brain, stomach, any tissue, including the brain. To grow a mini brain demands considerable skill in cell culturing, but, underline the but, the cells themselves don't need any guidance. We just let the cells do what they want to do. They already have the genes. They're self-directed. Okay? All they need is the opportunity. It's like really bright students may not need much in the way of teaching to progress, as long as they don't get in the way. By providing the right conditions to support and nurture the cells, the mini brain can self-organize itself, and build itself as an embryo does, but only so far. After all, there's no blood supply. It doesn't know its axis, what's up and down or side to side, so it can't orient itself properly. So is it a mature brain? No. It's a very early version of a brain. But still, it is a brain. And that's kind of what it looks like, but actually a better picture, this is the size. These little mini brains, I mean, they're a third the size of the thumb, a quarter the size of the thumb. So they're kind of the size of an early embryonic brain, okay? They can't go much further than that because they don't have any blood supply. And so it all comes from diffusion, and that's not a very efficient system. But even so, these mini brains actually look like what they could become adult brains. Now, this is Philip Ball commenting on this. I should be very clear what this term mini brain implies. Human nerve cells growing in the cell culture in this way cannot make a brain. But these nerve cells do start to create, under the direction of their own genetic programs, some of the features that a real brain exhibits. They become specialized into some of the many different types of nerve cells that are found in mature brains, and they also have an architecture. You could develop a neocortex, a layered neocortex. It looks surprisingly like a kind of an embryonic form of an adult neocortex with layers. And then he had some, I mean, he was a guinea pig for all these studies, so they biopsied him, moved over his shoulder to get some fibroblasts, reversed engineered them into his own little version of a mini brain. And so this are his feelings about it. There's no rule book to tell me how I should feel about my mini brain. I didn't lie awake at night fretting over its welfare. This mass of tissue made from my skin did not take on the status of an individual, but I felt oddly fond of the cells, doing their very best to fulfill a role in the absence of guiding influence from their somatic source, meaning the rest of the body parts and the brain. This was life in all its teeming, multiplying glory, spilling out from a pairing of me. That's a magnificent way of expressing it. This is kind of new life, been created from adult fibroblasts to turn it into a version, an embryonic version, of the brain. Well, now we're going to look at how this impacts on the difference between a Neanderthal brain and our brain. Turns out that genetically there isn't a lot of difference. There are a few genes that you can count them on the number of fingers in your hands with a few fingers left over. But a couple of those genes are really important. And the proteins they make may differ by no more than one amino acid. But it makes a huge difference to the mini brain that develops and might explain the difference between Neanderthals and modern humans. Well, just to remind you, they were found, the first skeleton, in the mid-1800s in Germany. They ranged all over Eurasia, lived anywhere from 400,000 to 30,000 years ago, and about 700,000 years ago, there was remember that one I showed you about illustrating the extinction event around 900,000 years ago? Well, move that clock forward about 400,000 years, and you've got the branching point here, with one major branch eventually becoming us. Other branches becoming ghost species, meaning no fossils were ever found, but there's the genetic mark of other genes, other species. And the other branch became Neanderthals and Denisovans. Okay? So, very closely related species. They probably did themselves in because they often lived in small groups, and that would certainly lead to inbreeding. And many of us, probably of European origin, carry around 1 or 2% of Neanderthal genes. And if we live in Australia or Southeast Asia, they might contain 4 to 6% of Denisovan DNA passed on. Well, then we come to this, how old are modern humans? Well, some people argue about this. I don't buy it. These skulls that were found in Morocco around 300,000 years ago plus were said to be the earliest modern humans. But boy, I look at this, and that's not what I see. It may be, but if you look at this has far more in common with Neanderthals, right? The big bony eyebrow over here, the sloping forehead, the longitudinally flat top of the head, the bulging at the back of the head, the so-called kind of bunion over here, and large orbits, large optic nerve holes. Now that's not the optic nerve hole, but anyway, meaning larger optic nerves and therefore probably more nerve fibers in it. And this robust jaw. Boy, the teeth are in awfully good shape, aren't they? No dentist has seen this one, so they must have eaten well without brushing their teeth or mouth washing or doing anything kind of thing. So probably, I don't know this, but this person's probably in their 20s, could be late teens, and pretty healthy, but closer, skeletally speaking, to I think, to what became Neanderthals than modern humans. Now here's kind of just introduce this. So we have these pluripotent stem cells, meaning they have the capability of diverging, multiplying, and differentiating to become a brain or other tissues. That's their capability. And even mimicking a neocortex here with its layered levels. It doesn't grow beyond a certain stage for the reasons I mentioned earlier, because it doesn't have a proper blood supply, it doesn't have a 3D axis, it doesn't have a good way of getting rid of its garbage, so it's limited in how far it can go. But back to the genetic story here. Here we are Neanderthals. So here's the protein made by this one gene that differs. And it only differs by one amino acid, lysine in Neanderthals, arginine in modern humans. Just one amino acid out of hundreds. And one gene makes a huge difference in the protein and the effects of that protein. Now they both lead to the development of multiple nerve cells, but more nerve cells in the case of modern humans. But you know, layered cortex, 5-6 layers in the neocortex, but more cells in modern humans. And so that's incredible that one amino acid in one protein from one gene makes such an enormous musculoskeletal behavioral difference. Isn't that amazing? I think it is. And so we move on. I've already mentioned these limitations of brain organoids, so I won't go through that. But here's where this field's going. This is now the creation of a human-rat hybrid brain. That tweaked your fancy?
No.
So, well, it might not, Jane, but think about it. If you're thinking about restoring function to a brain, if you can implant brain tissue from somebody with a hole because of a ischemic stroke or whatever, or somebody whose spinal cord has been traumatized, and you can fill in the gap, yeah, makes sense. These are early takes on this, so don't be too hard on this. People are right at the frontier. This is why I think at least 2 or 3 of these people are going to get a Nobel Prize, because they're right at the frontier. So what did they do? Well, they developed organoids, human organoids. You saw that little picture, the thing, what, 2 or 3, well, a couple of centimeters small. And they engrafted or grafted that in a very young 3 to 7 day old pup. Engrafted it in the brain. Well, it didn't just sit there. it actually developed, the neurons developed, they became larger, but they also set on processes right into the rat brain and made connections with rat neurons, and meaningful rat neurons, meaning you could stimulate the part of the brain here, show that there were connections throughout the kind of the rat brain, and that, as I said here, blue light, oh, by the way, they also engineered a protein into the cell here that if you shone a blue light on it, it would electrically discharge. So if you shone that little light, there's the blue light on this organoid, or the human organoid, neurons in that implanted organoid here would be excited, and their messages spread out into the rat brain, but not in a kind of a useless way. They actually form networks, functionally significant networks. That's extraordinary. You have two separate species, and one species, neurons, well, they're talking to one another. And they actually, some complex behavior is possible. So very early days here, but fascinating, I think. So let's move on. I mean, I could stop right here and the excitement wouldn't be over, but let's keep going. Learning change, yeah.
Excuse me, just before you move on, is it possible to take a human brain out? Somebody's died, you want to keep the brain, keep it alive in the lab by pumping blood?
No, we don't want your brain shooting.
I do. No, but I'm just thinking down the road because is it possible to keep the brain alive? That's the answer I want.
There's no theoretical reason why we couldn't do a little burr hole into your brain while it's still alive.
Right.
And grow it in a petri dish.
Yeah.
With these transcription proteins, and reverse engineer them into a much earlier version and grow many versions of you, which could potentially be implanted into whatever other sources.
Well, I mean, this is the question.
Yeah. So a little bit of Sheila could be living on.
Yeah, but it's quite fascinating because you think now of. I'm just looking at this to how we have iPhones and they are now part of our umbilical cord, and they're absolutely everywhere in the world. Yeah. And they're looking at the brain, and I mean, I'm looking down hundreds of years.
Yeah.
This
isn't going to take hundreds of years to go.
No, this would no.
This field is moving very rapidly. This is going to be within this century, and at the latest the next century. After all, we're 1924, or at least in 2024, and 100 years ago, we didn't know any other galaxy than ours, and not even that. There are a ton of stuff, a lot of stuff. Quantum mechanics was on the horizon.
Yeah.
Unimagined. So we're in the early stages, and there are so many more people well-trained around the world. I mean, what's the world's population now? 8 billion?
8 billion.
At the time of the kind of the early physics revolution I was kind of talking about, there were what, a billion? A billion and a half? We have so many more engineers and scientists and tools to play with and that kind of thing. I have no doubt the pace is picking up, for sure. This is not just a little tool for Europeans or high-profile American universities, for sure. A lot of the really interesting stuff, believe it or not, is coming out of China. So no surprise about that. They're now in the top 10 and the top 10 universities in the world for at least the quality of the literature that they produce. So that's a bit of a message, I think, we're
I
showing
mean,
us about.
all this leads on to manipulation, power, propaganda.
Okay, well,
Yeah.
it's not propaganda, this is international, no, this international group assessing the quality of these papers. It's not the Chinese evaluating them.
I don't mean propaganda, but I mean the implanting, God knows.
Yeah, well,
That's the whole story, yeah.
it is. Now, are there interesting, yeah, there are. It's very interesting, I mean, these are based on functional MRI studies where you could, where the brain kind of lights up, depending on whether it's active or not. And so based on that kind of relatively soft evidence, but still evidence, it's interesting what the effects of music are. Because there's quite a difference in the activity in the brain between somebody who's playing the music and people sitting in the audience listening to the music. Because people listening to the music, you can see the activity in areas of the brain where you would expect it. The auditory cortex and some of the connections to that auditory cortex. But the person playing the stuff, the whole brain kind of lights up. And so it's, from a neural point of view, it's quite a different experience. And sometimes you can show the people who practice a lot that the neocortex thickens. Now, not because they've got more nerve cells, but because the nerve cells that are there have more processes, more connections with one another, and they're probably thicker and more efficient. And no surprise, after this last 3 weeks, these changes are most pronounced in the young. So no surprise there. Now, what about regeneration in stem cells? Well, blood and skin, well, we know. I mentioned this. If you cut the skin, within a few days, you've got a little scar. 2 or 3 weeks later, you've got a little pink thing, the scab falls off. You can see that there's new skin there. Well, that's been formed from stem cells in the basal layer of the skin. Okay? So you can actually see the effects of stem cells. But the same happens, say the liver is replaced every 3 years. I don't know how they ever established that. And certainly rebirth of muscle fibers, that's encouraging for stroke. But the brain, no. Maybe for the hippocampus, but that's questionable. One of the problems with stem cells is that they're just as susceptible to the natural injuries of aging as other cells, and they tire. They actually more mutations. They accumulate epigenetic markers, which I talked about in week 2. Telomeres get shorter. All of that stuff that you'd expect. The mitochondria is functionally competent. And so the stem cells start to fail. If the stem cells start to fail, the whole thing starts to fail because there's no replacement for some tissues. And furthermore, if you get one population of stem cells that seems to have found one line that it's kind of dedicated to, it crowds out the other stem cells. So everyone suffers because one gets its way. And that's what happens in cancer, doesn't it? One cell, one version of the cell, crowds out its brethren, and sometimes with bad results we know about. Now, here's where this comes in. Imagine this. This is where the Nobel's going to come. Now, this is in situ. This isn't out in a petri dish here. Reprogramming cells to make neurons could be used to repair spinal cord or even brain damage. Well, when I see a sentence like that, that reads like a sentence in a grant submission, where you really want to say, gee, we're doing really interesting, great stuff. And still, at the University of Texas, Zhang showed that, well, you know the brain has three types of cells: glial cells, which are kind of support cells, microglial cells, which have immune functions, and nerve cells. What his group managed to do in the adult, in situ, in an adult, is using transcription factors, change some of those support cells into an early version of neurons, which became neurons. That's amazing. That's totally amazing. I mean, that's the kind of thing, say, in Parkinson's disease, that you'd be really, really interested in. Because so much of the pathology is so well focused that if you were ever going to use implants, or stem cell implants is a great place to do it. The problem with the nervous system is this: it's not enough to have a kind of a destroyed area and then bring in a bunch of immigrant cells and hope the whole thing's going to work. Because unless those immigrant cells, the stem cells, form meaningful connections to their neighbors, there's no point in the exercise. I mean, that's how the nervous system works. It's connected. There are all these complex networks. And in the peripheral nervous system, it's far simpler, kind of some version of reestablishing networks, but in the central nervous system, it's a far more iffy matter. Well, if that kind of system works, well, why can't we deal with macular degeneration that way? Why not just replace the retinal cells? Why can't we transform them in vivo? Makes sense to me. And why can't we replace, I mean, the biggest source of deafness in our age group is in the hair cells in the ear. Hearing aids don't help them. You actually have to rejuvenate the hair cells. Where would they come from? From some approach like this. Regrow them, repopulate. And so that's clearly a target. Now here's a slide I've showed you before, but this is a different take on things. Now we're getting into neural or computer-human interface stuff. That's a huge field that's really taking off now. And when we did the series on AI, we talked about some of this stuff. And we'll refer to some of that today, because it's really very promising. Well, what happens, this is the macaque. I used to have macaques in my lab 11. I would never do that again, but anyway, did. So here, an injury was created in the spinal cord, so that the legs are disconnected from the brain. So we have paralyzed, paraplegic. Yeah, the rhesus monkey is paraplegic. Paraplegic. But what if you could take the information in the brain, the command signals from the brain, and directly bypass that whole area and connect it to the assembly of neurons in the lumbosacral cord? At first thought, it's an intimidatingly complex system, the nervous system, and my first thought would be, oh, that's an overreach, but you know, it works. And so the signals being recorded here and analyzed here with the help of AI, back in the AI business, there's an intrinsic organization. The lumbosacral cord knows how to walk on its own. But to make it walk in response to the brain telling it what to do, you need a connection between the two. Here's the connection between the two. So it surprisingly works. And if that catches you, how about this? This is from Switzerland, a group in Switzerland, read it. And a very enterprising group. Now, you can imagine that this person, a man, had a traumatic injury involving his cervical cord, the lower cervical cord, and he couldn't walk. I'm skipping an intermediate stage in here, right up to the most recent paper in Nature a couple of years ago, so I know they're well beyond this. Here is there are grid of electrodes here over the leg areas here, so recording neural signals from the leg. So the intention to walk, the voluntary intention to walk, that signal is being recorded here, or some crude version of it. AI analyzes that material, and then those signals are sent, like the macaque, down to the lumbosacral cord, bypassing the trouble in the lower cervical cord, just like the macaque. This fellow's been walking independently. He's got a backpack. And earlier versions were not nearly as successful. But with this approach, he can voluntarily say, gee, well, I wouldn't say that, but well, it might. But anyway, the impulse, he might have the impulse to walk, and it will translate to a sequential movement of his legs that's appropriate. And the extraordinary thing about it is it actually works on uneven ground. That is very promising. So the correction here doesn't have to be nearly as complex as the neural system. Understand that? For success. You don't have to have every little thing in place. Just close to it. Enough to make it work. And if you were impressed by that, this blew my mind. Just this year, August the 15th. And you know that I had helping out a fellow in the 60s in town with this disease. And he, ALS, and he lost his speech, meaning his speech became incomprehensible. And he was a very social person, very outgoing, and very early in the game said, gee, if that happens to me, I want Maine. So he had Maine. And, but he didn't, and I didn't know about this. Now I can tell you that over the last 10 years especially, there have been very similar attempts to reestablish speech. But the earlier approaches, say 10 years ago, if I go back 10 years ago, subjects, even with the best technology at the time, the vocabulary was no more than 100 words, maybe 10 words a minute, with an error rate of around 30%. Not very meaningful. This vocabulary is thousands of words, an error rate of around less than 2%. Thousands of words, around 2%, and the speed is up to maybe half of our speed. Wow, that's transformative. If that had been available for our fellow here, that would have really been a game changer for him. And why isn't this so available? Well, it's because there are only a few centers now with the technology to make all of this happen. But you know, just looking at the past, that these are trials to sort stuff out, and it'll become easier and simpler, maybe not less expensive, but easier. So isn't that amazing to get around? Oh, and the thing, one of the differences here was a lot of the emphasis, and all of the emphasis early on, was based on the idea that we'll record the electrical signals related to speech, the kind of the words, the syllables of the speech. But they were clever enough to say, well, yeah, okay, that's part of it, but there are a lot of other things happening to make speech possible. So all of the articulatory muscles in the larynx, the pharynx, the tongue, the facial muscles that are actually responsible for making speech happen, those signals should be included, and that's what they included. Not more recording sites, but a broader swath. So it's a huge change, and so still clunky. You know, we're still restricted to kind of special centers, and still not kind of a mobile getting around. But as far as speech goes, this is clearly where things are going. And this is just an example of neural, in this case, brain hybrids. Because that's where we're headed. This is the 1924 moment of 2024, right? Early in the game. Early in the game, and with the potential to really go somewhere. If they enjoy more success with that speech thing and it becomes more widely available, there's no go for that for sure. So what, if we start to put some of these things together, it is possible, as I said, to grow organs by reverse engineering mature, well-differentiated cells into cells that have the potential to become any cell. And here, just down here, evolution by natural selection, which has driven the emergence of millions of species, may be superseded by human control within this century or the next. That's a genuine, for some, fear, and maybe justly, but clearly the tools are there. With, I mean, yes, there was a Nobel Prize given for gene editing back in, 2020? And, but that technology has moved at a blistering speed just in the 4 years since then. And so it's now possible to not just edit one gene, but possibly hundreds of genes. And behavioral changes come with hundreds of genes, potentially. Yeah.
I'm curious, if they ever develop to be able to grow a liver or grow a kidney from your own cells, would that take care of the problem of rejection from your body?
As long as you suppress the immune system or find a way to do it. That implant into the young rat brain was done to deliberately circumvent the immune system. Like peanut allergies, that kind of thing. If the kid is, the baby, or the kid is exposed to peanut things really early, most of them don't develop an allergy. We've been too nice to people. That's what happened with polio, because sanitation came along, so kids weren't exposed to the virus very, very early in life. And then we had these polio epidemics, which were a regular feature of my childhood. So it's a bit of a knock on cleaning up because of overcleaning and living in homes all sealed up, kind of thing. So anyway, we're then the sentence up here. New parts for old. Well, now we're at the point of 3D printing of arteries, veins, heart valves, and growing organs inside or outside the body. Well, that's where we're at. And my grandson in Alaska, 15, just used 3D printing to create his own personally designed clutch for an off-road vehicle he's designing. So boy, things have changed. Because technologies. Here's the last one I'll put up. Young blood to stay young. I mean, this sounds like Frankenstein, right? And it is, but after all, what is blood? It's a perfusate that's going around. It touches every organ. So any proteins that are released or other substances get into the circulation. That's how picking up cancer is possible, by analyzing the blood, because a little bit of the abnormal DNA gets in the blood and you can identify it. So it looked very promising a few years ago, and still is. So no surprise that there might be some compounds in the blood that might have a benefit. So what's the evidence? I'm always distrustful of studies in mice. That young blood restores muscle and liver cells in older mice. Well, great for the mice. Young blood given to older animals improves the latter's memory. Well, okay. But here's an interesting one. Proteins in umbilical blood revitalize the hippocampus memory part, and one protein in older blood given to younger animals impaired the latter's memory. So it suggests that yes, there are active biological compounds, most of them proteins, in the blood that do have an effect. Here's an interesting thing. Treating young mice with old blood quickly increased the number of senescent cells. Well, that was lecture 2, or maybe lecture 1 about senescent cells, kind of cells that are no longer capable of dividing. And then I thought, what about transfusions of blood? Where did the transfusion donor? Were they old? Does the transfusion, if it came from us, would it be less effective, even deleterious, because of proteins in that blood? Well, no one's thought about that. Or put another way, if we got our transfusions from kids, well, I can't think of kid donors, under 10, but potentially, if you think about it, there might be a benefit. So the point is, some proteins in younger blood can reduce the effects of aging, and the reverse. Older blood has factors that lead to premature aging. I don't think there's a surprise in this. I mean, all this molecular chemical stuff floating around in our blood is it's been hardly analyzed so far. So I think it probably does have the potential. Now, I'm not going to go over that because you know all this stuff. Well, this is my take on how do we make the best of our lives. Being grateful and generous helps. Certainly fostering relationships and expanding them to bring new interests into our lives to offset the all too familiar inevitable losses of friends and partners, kind of thing. Eat right, drink less, exercise right, and good genes. And I mean, that's all common sense. This is my kind of take on the whole thing, so it wouldn't be Bill Brown without a slide on perspectives. Species evolve. I'm not talking about theology here, just the science. Species evolve. No species lasts forever. In the human story, no more than usually 500,000 years. I know that sounds like a long time, but it isn't from an evolutionary point of view. And often less than that. No life form is immortal based on biological evidence. Human life may be extended by technologies, but in the end there is an end, biologically speaking. Now, we don't really know what's possible for the length of humans' life, I don't know. But in my own view, just looking at my own life, is that the quality of life is more important than longevity. And for a species that often claims some version of immortality, we seem to spend a lot of time and energy trying to stay alive. It's almost as if we didn't really believe what we were claiming. And from a science perspective, the only immortality that I can think of is in the laws of thermodynamics or energy. And energy is neither created or lost. It can become useless or inaccessible, but entropic, if you like, it's a scientific word for it. But other than that, stars come and go, compounds come and go. There were no atoms and particles in the earliest part of the so-called Big Bang kind of thing. They were all created in that moment. So none of those things are immortal. And but they might be recyclable with multiple universes. I mean, that's possible. That's kind of well. But you see why, I hope you see where the last one here goes. You can see why I got quite turned on in looking at this material, because I realized just how revolutionary this material is. And it was so unanticipated, it kind of crept up on people. It's interesting that with Jennifer Doudna and Emmanuel Carpentier in their gene editing one, so that's only 4 years ago, in their books, make no mention of this stuff. And maybe they weren't interested. But it's clearly a field that has enormous potential. You can see where this is going. You might not like where it's going, but it will go, because there is no universal government or restriction on any of this kind of stuff. And so it's a good thing to think about, are these directions that we would support, or can we see hope for, or use for humanity and other species. After all, we're busily destroying species by the millions. We are not the most important species. We'll be out-survived by bacteria, archaea for sure, and our own species. Well, if we follow the, just look at the several species that led up to us in just a Homo period, 2 million years of our history, every one of them came and went. Is there any reason to think that we'll be any different than the rest? That would be a radical thought, to think that we are the immortal ones as a species. That doesn't make any sense at all. And we do know from an evolutionary point of view, just looking at the genome, that it's actively changing a lot. And so things are changing. Some of it not good, in response to environmental changes and other changes. So if we revisited Earth 500,000 years from now, are we going to find versions of ourselves wandering around? I don't know. My own personal hope is, I hope it won't be us. It will be some more evolved version of ourselves. Hopefully brighter, hopefully more responsible, but we don't know that. I do know that our species has been the most destructive species in all of evolution that I can think of. I can't think of another one that collectively has done more damage to other species as well as one another. So we may be the most intelligent the species, but we haven't been the wisest. And we seem incapable of collective action. And just look at the Middle East these days. You need no more proof than that. So I think this is really interesting stuff. I hope you found it that way too. So next week we do the Alzheimer's disease thing and the dementias, and that's an important one because there are new drugs out, controversial ones and very expensive ones, but it's important to have an update on that. And then the Nobel series starts in November 6th with physics and chemistry then medicine / physiology, and then Peace Prize, and economics prize, and then Bell made is the closer with literature. And so that's what's ahead. So I've enjoyed all of this. I hope you have too. And it's kind of mind-stretching stuff. But I do think the evidence I've presented here is actually good evidence. So any questions, any thoughts? Yes?
If you have stem cell therapy or whatever it's called,
Yeah.
where do the stem cells come from?
Well, some of them actually come from other species. And,
Oh, okay.
and some from here. You know, there was a period, stem cell therapy as an idea goes back at least to the 1990s.
Right.
And with the hope that, for example, I remember a church we went to in Newton, there was a young man with ALS and his younger brother, both graduates of MIT, turned his whole career around, raised billions, was on 60 Minutes twice about this kind of stuff, and his brother was one of the first recipients of stem cell therapy. It was being pushed by a group at Johns Hopkins, and has been tried in a number of sources. And for example, in the brain stem for Parkinson's disease, early days. Actually, the first studies were done in Mexico.
Well, my grandson is super autistic.
Yeah,
And my stepdaughter's my step.
yeah,
So my stepdaughter went down to, and I'm not sure if it wasn't Puerto Rico or Mexico or something, and paid an enormous amount of money at one of these health things for stem
Yeah.
cell things. So I was interested where the stem cell. I don't think it did anything. I mean, who knows? I don't know.
I think people stayed out of the sun, they wouldn't need stem cell therapy for their skin.
But I think this is a bit of a propaganda thing.
I think it is.
Yeah.
It's hype, and it's created a lot of money for some people.
Well, it's interesting because it's marketed as a package.
Yeah.
They arrange the hotel, they arrange absolutely everything. And I mean, I disagree with the mother, but I can see her search for anything for
Right.
the son, and it's not going to be found. But I mean, it's so well marketed and packaged, and you get the whole thing, and they meet you at the
Yeah,
airport.
it's sexy. It's sexy.
It's sexy.
that weird.
Exactly.
It would be big on YouTube.
I haven't, I mean, all I know is from this personal
Yeah.
experience.
Yeah. But the idea is not a wrong one. If you had the right stem cells, and yeah.
I was going to ask that about the stem cells. Have they shown that stem cells from me to replace something here are better than stem cells from another person?
Stem cells from younger donors are more effective than stem cells derived from older people. That's true, for the reasons I mentioned earlier, because if you take the stem cells themselves age and become less effective. But stem cells, I mean, there are stem cells and stem cells. If you think about it, the original egg with one cell, that's one stem cell from which everything, including the placenta, came. To much more differentiated stem cells whose only job is to crank out red blood cells or certain immune-related cells in the bone marrow. So they differentiate. But they can be redirected back. I mean, that's the revolution, that you can actually reverse engineer. If you think about this, it's an amazing philosophical, because this is science fiction stuff that people kind of talked about, hundreds of years ago, with the possibility of new life from older tissues and a hope that people. or fighting a young wife might be kind of re-engineer you, so to speak.
Mr. Murdoch's done it many times.
Yeah, many times. So this idea that you can kind of kickstart youth by associating with youth, or in some way borrowing cells from them, is it's a very catchy idea. But it does have some physiological and molecular basis for thinking that. It's not a crazy idea. The applications. So we're in early days here. This is we're in the early days of the 20th century or 21st century compared to the last century, just like that, I think. So the importance now is to establish as high a standard as we can for the quality of the work being done, recognizing that there are going to be lots of false leads. But I'm surprised at how daring some of the investigators were, just going back, 20, 30 years. I mean, who would have paid money to kind of try to re-engineer a frog or a tadpole? I mean, who's going to pay any attention to that? And, well, they didn't. But fortunately, science in those days was cheap. And they were using animals that were easy to work with. It becomes very expensive when you get up to the macaque, rhesus monkey stage, and less palatable to a public, and deservedly so, I think. Although it's interesting that during COVID, there were restrictions in the United States by law that weren't allowed to experiment on chimpanzees in place before COVID. As soon as they got into COVID, and they needed a model close to humans to try out some of the stuff, on the quiet, chimpanzees were re-included. Not many people know about that. And a lot of that stuff is done over in the west, in Montana, beyond sight. So it's, anyway, I do think it's not a surprise that the whole central idea of this, if you think about this, if you think about development, is the one track we're, in this room we're familiar with, is that one sperm, one egg, two cells get together and seemingly magically over days and weeks and months specialize increasingly and establish relationships with one another and took on separate jobs without changing the genome. How did that happen? By selectively blocking certain genes and allowing others to work. So in other words, if you were destined, say, to become a nerve cell, well, any genes in your genome that in any way are related to what might have become a kidney cell or a liver cell or whatever cell were all turned off. So think of a highly differentiated cell as a genome, and if you could actually see all those tags, those methyl tags, or histone tags on the genome, you'd see a very complex network of keys, silencing some, turning others on. That's what's different. The genome is not different. It remains the same. That's why you can reverse engineer it. That's why you can take columnar cells out of a tadpole or a frog that are fully differentiated into an animal form, doing a very specialized job. They're not doing any kidney functions, they're not doing any brain functions, they're just doing what a columnar cell lining a particular area of the bowel does. That's what they're doing. because all these other genes have been turned off. Follow what I'm saying? Very selectively. So to turn that around, all you need to do is pull all the tags off. That's what transcription factors do. They're very good about when, for example, everyone in this room, for all the cells, the differentiated cells in our body, and because of our age, every cell has a certain signature. Same genome, plus a lot of mutations, but same genome, all the cells kind of marked for a certain destiny or job that it has. But with the sex cells, the sperm and the egg, almost by magic, all those tags disappear. They disappear. All the tags come off. So they're pluripotent or tetra, okay? Same genome. And that's the amazing thing. That's what people didn't recognize.
What chemical controls the changes?
You're asking the God question.
That's the thing.
Yeah.
Why does it
Well,
happen to?
Well, that's a very fundamental and important question, Elizabeth, and I should shut up at this point, but I won't. Well, I don't know, and as far as I would ask the same question. It obviously is far more complex than we ever imagined. You think about when the human genome, when people, the Clinton's era, when it was mapped, a very incomplete version, I might say, after 10 years of work. And a lot of people were slapping one another in the back. Oh my God, we had the code. But it was never just the code. There are two codes. There's a code of the genes, that roadmap of genes that we have, and a whole other system that's barely been touched. Other genes, no doubt, and some of them are proteins and RNAs and that kind of thing, but you're correct. Where'd they come from? And, well, we don't know, because this is like an orchestra, like no other orchestra we've ever listened to, because there are a lot of instruments, not just the 30 in the orchestra that we go to listen, that kind of thing. There are millions, possibly billions of variations on this song possible, and we don't know actually where that comes from. We don't. And neither was, as I say, the gene editing stuff and the 2 books that the women published at the time, they don't even touch on it. Yeah, they didn't touch on it because there wasn't much evidence for it. But it's clear that that's what's happening. And so you think of yourself aging, you're accumulating tags. That's what you're doing. That's part of it. It's happening. You're accumulating tags. And every subsystem in your cell is independently accumulating its draggy tags. And so it's kind of a total system collapse, but not all simultaneously, fortunately. And so that's why we have gray hair. That's why we developed a lot of the system problems that we have. I mean, sure, trauma, chance, all those things play a role. But overall, the general pattern of aging is very similar, isn't it? Even if the piece isn't the same, and how it affects people. So you can see why I got really, I mean, that's a very important question, that it is. But we're early in the century, 24. We have 76 more years in the century to fill in the blanks. And just imagine how far we've come in the last 100 years. a long, long way. So, and none of us will be here when other people know that. If they stop belonging. Okay, all right, well, listen, thank you very much, and we'll see you next week, I hope.
Yes.